All right, welcome you guys to Anatomy and Physiology for Speech and Hearing. I'm Shanna Stack, and I'm going to cover Chapter 10, Physiology of Hearing for you guys. Here are a list of links that are embedded within your guys' PowerPoint. To begin, I'm going to give you an overview or an introduction to physiology of audition. In general, the auditory mechanism can hear 10 octaves, being 20 hertz to 2,000 hertz. Okay. There are four different tasks that are required in terms of hearing. One is to identify small changes in frequency, so changes in pitch. Identify small changes in amplitude, which is the intensity of the sound. Differentiate a signal embedded within background noise, so be able to weed out the important information. So, for example, when you're at a restaurant and you're trying to listen to your friend talk, the ability to really target whatever that signal is, and then interpret rapid sequences of different sounds. All right, so this is an overview, but in general, the outer ear collects the sound and then somewhat shapes its frequency components. So we'll talk about how that shaping occurs. The middle ear matches that airborne acoustic signal. So acoustic signals are basically changes in air pressure that travel through that external auditory meatus, right? And then cause that tympanic membrane to vibrate, and then send that vibration through those ossicles to that oval window. So it matches that airborne acoustic signal with a fluid medium of within the cochlea. Okay, and then the inner ear performs a temporal analysis of the acoustic signal. So it tells you when that sound came and in what order and for how long, and also a spectral analysis, being the frequency and intensity, or the pitch and the loudness of that acoustic signal. Once that's been done, the auditory pathway is going to convey the signal to the brain and brainstem, and then additional processing is going to occur at that level of the central nervous system, and then the cerebral cortex is going to interpret that acoustic signal and bring it to conscious thought. All right, to begin, the outer ear collects and shapes the sound, and it does this through selective enhancement. So the outer ear has resonant frequencies that are more amplified when they reach the ear, and non-resonant frequencies that are rejected when they meet the ear. So they either fit within that the pinna and the external auditory meatus, or they don't and they're kicked out. So 2,000 hertz is selectively amplified at the highest gain, being that you're going to get the most bang for the buck in terms of 20,000 hertz at a 45-degree angle. And so we'll talk about that one specifically. The pinna funnels the sound right to the external auditory meatus, and then the external auditory meatus is the largest contributor to the selective enhancement. And it allows that 2,000 hertz to bounce off of its walls, basically. And 2,000 hertz is just this cycle per second, right? And so it has the shape of that frequency, the basically that cycles per second fits within if you think of a wave, that wave fits within the external auditory meatus, okay, in a way that is amplified. So it creates more cycles per second, okay. So you get a total gain of approximately 20 decibels. So, and decibels are not on a linear fashion, so 10 dBs is not twice or half the size of 20 dBs. 20 dBs is on a logarithmic scale, so it's exponentially higher than 10 dBs. Okay. All right, and then we move to the middle ear, and the middle ear job is to relay this acoustic information in the terms of air pressure change. It transfers it through kind of this mechanical process where the ossicles vibrate in a mechanical way and create force against the oval window, and that then moves the fluid within the inner ear. And it does that in a number of ways. I'm going to tell you about it on this next slide. So it does this via this impedance matching function. So it's much harder to move fluid. It takes much more force to move the fluid in the inner ear than it does to move air in our atmosphere. We can create air pressure waves with much less force than waves in water, for example, if you want to think about it that way. So in order to overcome that and not lose that acoustic signal because we can't seem to push that fluid, we've evolved to be able to we've evolved this impedance matching function. So and we do it via three mechanisms. One is area. The second one's this lever action, and the third one's this buckling action. So in terms of area, we have a very large tympanic membrane and a very tiny oval window. So just by taking that large area and all the force that's exerted on that very large area and funneling it down to a smaller area, we get more force per area, right? So which means more pressure. So that's one way to get more pressure on the oval window to push that fluid. And then we also have this lever action, okay, where the manubrium of the malleus is longer than the lenticular process of the incus, and because of that difference in length, just think of it kind of like the difference in area. It's not exactly the same, but it's a way to think about it. The difference in that bigger area being that longer length being funneled down to a shorter length gives us more force, which then is turned into this extra 2 decibels of gain. So you'll see that the area is the biggest impedance matching function. And then lastly, we have this buckling of the tympanic membrane. So the tympanic membrane doesn't move equally throughout the whole TM. So where the head of the malleus is embedded within the tympanic membrane, that part of it doesn't move as well. Okay, and because it doesn't move as far, all of that force that would be used to move the tympanic membrane really far, like the rest of the tympanic membrane that doesn't have the head of the malleus embedded in it, all of that force is then transferred into moving the vesicular chain. So that gives us another 4 to 6 dB gain. All right, now we get to talk about the inner ear, specifically our mechanism for balance, the vestibular mechanism. Then we're going to talk about the auditory mechanism in twofold, so mechanical and electrical events, and then finish up by talking about auditory pathways. All right, so we have gone over this in the last chapter. So the vestibular mechanism is in charge of balance. So it tells us where our body is in space. And it does that in 2 dimensions, in terms of acceleration and in rotation. And I've listed here the sense organs responsible for each. So the saccule and neutricle give us information about acceleration, whereas the semicircular canals and the ampullae that are associated with them give us information about rotation. All right, now we get to move to the mechanical events of the inner ear. Okay, so and the inner ear again is responsible for temporal and spectral analysis of that acoustic signal. So in general, so this is sort of a recap of all the things that happen. So we have air pressure changes in the environment, and that's basically we're taking an air pressure change and turning it into some sort of meaningful information that we're using. So we have air pressure changes in the environment. The outer ear funnels and selectively amplifies the acoustic signal. Then the middle ear receives that and also adds those 3 impedance matching functions to help push that oval window into the inner ear and displace the perilymph and the scala vestibuli. Okay, and then that displacement of the perilymph in the scala vestibuli displaces that Reisner's membrane. And remember, Reisner's membrane is the membrane between the scala vestibuli and the endolymph, or that scala media. Okay, and when Reisner's membrane is displaced, it displaces the endolymph in the scala media, which then displaces the basilar membrane, which is located in the organ of Corti, which is that organ for hearing. So those are all the mechanical events that happen in order for this acoustic information to be gathered. All right. So continue. We're going to continue talking about the mechanical events. So so when we talk about the movement of this perilymph and this endolymph in the cochlea, we kind of talk about it in terms of this traveling wave. Okay, so it's this periodic vibration of the fluid being displaced by the oval window and then translated to that basilar membrane, where the basilar membrane is being displaced, and then the hair cells are being moved, and then that eighth cochlear nerve is then picking up that information. So the point of maximum excursion or perturbation of the basilar membrane really varies depending on the location in the cochlea and the frequency of the acoustic signal. So closer to the vestibuli, you're going to have a thinner, tenser, and less massy basilar membrane. Okay, and closer to the apex, you're going to have a thicker, flaccid, more massy basilar membrane. So, so there's this tonotopic relationship or arrangement of the basilar membrane. So closer to the vestibule, so closer to the middle ear, you're going to be able to interpret higher frequency sounds. Okay, and that has to do with this having it be a thinner, tense, less massy structure. If you think of the vocal cords and how women have thinner, less massy vocal cords, so they typically have a higher frequency sound, the same principle is true for the basilar membrane. It's interpreting those higher frequency sounds, whereas closer to the apex of the cochlea, which is kind of counterintuitive because you think, thinner would be up higher, closer to the apex, but that's not true. So closer to the apex, you're going to be able to interpret those lower frequency sounds due to that thicker, flaccid, more massy basilar membrane. And so that brings us back to this point of maximum excursion. So you're going to get a higher maximum excursion with a more flaccid basilar membrane there closer to the apex. So go ahead and watch that video, and it'll help bring things together for you. All right, so then we're going to talk about this shearing action. So it's displacement of the hair cells in the organ of Corti relative to that tectorial membrane that sits right on top of it. Okay, and then we have the greatest point of maximum excursion of the basilar membrane. All right, and then the outer hair cells are embedded in the tectorial membrane, but the inner hair cells are not. So the inner hair cells are largely largely rely on the movement of the endolymph for that shearing action. All right, so then we have the stereocilia, and it's cilia located on the hair cells. And go ahead and watch this video, and it'll put together this whole depolarization, repolarization of the hair cells and the resting membrane potential and potassium channels. And we'll go through it, and I'll draw some pictures and everything for you guys in terms of what these neurotransmitters and calcium channels are, and what action potential and so forth. So watch this video, and then since we're and then I don't think I'll test you too hard on this until we go over neural, but do start learning it. All right, so we're going to talk about the ascending pathway, and we spend most of our time on the ascending pathway, and then we have just a little bit on the descending pathway. So this is how information is transmitted from the cochlea to the cerebral cortex. So to begin, so this is all brainstem here. So if you go to the next page. Okay, so here is our brain, and this is our brainstem. This is part of a brainstem too, actually. So basically all inferior colliculi and down are brainstem, and this is your ear. Okay. So the cochlear nucleus, and you'll see it's set up bilaterally. The cochlear nucleus relays um, the the information in regards of time, frequency, and amplitude from the cochlea um, up through the cochlear nucleus, and then as that information is processed, that temporal frequency and intensity is all processed here and then sent to the superior olivary complex right here. But also, there's something kind of tricky that happens at the cochlear nucleus. We also have that information travel over to the other side of the brainstem. Okay, so now we have information. Say this is the right cochlea. We have information from the right cochlea headed over to the left side of the brainstem and to the right side of the brainstem. So both sides of the brainstem at this point, both pathways are receiving information from each cochlea. Okay, so there's some decussation or crossing over of that information. So once the information reaches the superior olivary complex, um, it's processed in terms of that information is then processed in terms of understanding or localizing where that sound is in space. And and that's important, and this will help you remember it. You need to have information from both cochlea to be able to interpret where information is coming from in terms of where from which direction. So you need two ears. You need binaural hearing to be able to localize sound, and you need that you need to be able to process it on both sides. So we need this decussation to occur, okay, in order for that sound localization to happen. So think binaural bi. We need two cochlea on board, and we also need both superior olivary complexes on board in order to localize the sound. Okay, so right here we're relaying and enhancing time, frequency, and amplitude. Here we're localizing sound. Okay. And then the inferior olivary complex is the first time that we start to inhibit any auditory information. So any of that background information that's not all that important. It helps organize the information in terms of time, frequency, and amplitude as well. So this is sort of the first shut off place, and in terms of this is background information that we don't need to send up for conscious awareness. Okay, and an additional organization of it. So here this is just relay and enhance. This is the first place where it's being organized and some of it's being sent back. Okay, so first place of organization, inferior um, inferior colliculi, and we'll look at where the inferior colliculi is when we get to neuro. It's really cool. You can see it really well. It's pretty dang awesome. So then we move up. Oh shoot. We move up to the medial geniculate body, which is right here. So now we're within the brain. All this is brainstem stuff below. So here we're within the brain, and this is really just a relay point to the thalamus. Okay, and I don't know if it's showing the thalamus. It's not. So the thalamus is right up here, and maybe on that next page. No. Okay, so the thalamus is really just a relay. It's kind of like the bus station for all of these different neural pathways in the brain. And it's not showing it on this picture or the next one, but so just think this thalamus is just kind of a relay station. So the medial geniculate body is just a bus on that relay system. And it heads to the thalamus to ask the thalamus, where should I go? And then the thalamus says, you need to go to the cerebral cortex, which is up here. Okay, and actually the temporal lobe of the cerebral cortex. Okay, and so the temporal lobe is just, if you remember your cranial bones, your temporal lobe is just the part of your brain right below the temporal bone, and that's where interpretation of the acoustic signal happens. That's where conscious awareness of that sound occurs, which is right there. So pretty cool stuff. So here's another picture to help put it together for you guys. So here's spinal cord, here's brainstem, this is your cochlea, your semicircular canals, middle ear, outer ear, this is your external auditory meatus, your pinna. Okay, so cochlea here, and then we go cochlear nucleus, superior olivary complex, inferior colliculi, medial geniculate body, and then actually goes thalamus, which is right here, but that's fine. You can just go with this. And then the temporal lobe. All right, so in summary, I wrote it out for you guys. The cochlear nucleus processes and enhances information. Superior olivary complex, and it also is at that point of, let me show you, the cochlear nucleus, that's where the point of decussation occurs. Okay, so that crossing over the sound. Superior olivary complex gets information from both cochlea, which helps with localization of sound. Then the inferior colliculi, that's the first point of weeding out what's important and what's not and organizing the information. Before that, it's, at the cochlear nucleus, it's being processed and enhanced, but no kind of thought or organization is occurring until we get to the inferior colliculi. So you can think, you have that enhanced signal, and you can localize the sound before you get any inhibitory response first, okay? And then inhibiting any of that sensory information, that sound, okay? And then it's being organized in terms of the time, frequency, and amplitude, and then travels to the medial geniculate body, which is just really a bus to send that information to the thalamus, and the thalamus then is the bus station sending that information to the temporal lobe, specifically the auditory cortex, where that acoustic signal is interpreted in a conscious way. So remember, decussation is that crossing over of the neural pathway. And there's another link for you guys to watch. All right, and then I'm just going to spend a little bit of time talking about the descending pathway. So if you remember, if we go back up to the inferior colliculi, okay, that's a, it kind of plays a role with this descending pathway. So the descending means it's information moving away from the brain and spinal cord. Okay, so it's ascending is sensory information heading out, descending is efferent motor information heading, or sorry, ascending is heading in, descending is heading out. So its primary role is inhibitory, and it just determines where that information should go. So should it go to the frontal lobe? Should it go in terms of really being aware of that information, or should it go to the posterior temporal lobe for attention? So all you need to know about the descending pathway is that it sends information where it needs to go, and it plays a role in inhibiting that sensory information when it needs to happen. All right, well, congratulations, you guys. Chapter 10 is done.